morning, Chemsky. You present yourself?
Hello, my name is James King, and I'm a designer. And I'd like to start by saying I fully agree with Alexandra's point that synthetic biology is definitely an area of design. And although I studied design for my education, I think that people that have studied engineering and science and then become synthetic biologists are just as much designers as people who want to ask, because what they do is a creative process. Having said that, my talk is called Making Design a Part of Scientific Practice. And that's what I think has been very interesting about the last few years in science and design. There's been very much a collaboration between people like myself, designers, and people like Alexandra and other synthetic biologists, as well as scientists in general. I'd like to start with a project which I made in 2006, and it's called Dressing the Meat of Tomorrow. There's a real technology that allows you to take a small sample of animal tissue, just a few cells, and grow it separate from the original animal's body to form an edible piece of meat. So the idea is that rather than growing an entire cow, you just grow the steak. The technical term for this is called in vitro cultured meat, but some people call it victimless meat because no animal has to die in order for us to eat meat. So let's imagine a future where this becomes the most normal, the most sustainable, the most humane way of producing our food. And then let's ask a few questions. So what size would it be if it's not limited by the size of any animal? What shape will it have if it's not limited to the anatomy of any animal? How much would it cost? And who would buy it? Would we be happy eating something that looked like this? Perhaps not. This is what it would look like if you took it from the Petri dish today and put it on someone's plate, except these pieces here would be about 3 centimeters wide. Recently in the press there's been talk of the $100,000 hamburger. Have people seen this? So in the last few years it's become, I guess more people are kind of investigating this area of food production. $100,000 is still too much to pay for a hamburger, and there are very good reasons why it costs that much money, because it's very, very difficult to do this type of science. What I was interested in is, in the future, what would happen if a chef, like, I'm going to come up with an English chef, Heston Blumenthal, so maybe Erling's, like a very famous chef, would become interested by the anatomy of these old animals called cows that no longer exist because no one eats them. And he's in the library, he's looking through these old textbooks of these anatomical illustrations. So this is a cross-section through a cow's stomach. And he selects the parts he finds most interesting, so not the boring parts that we eat today, but the more intricate and beautiful patterns in the abdomen and the brain, because he's just seeing these pictures and he's thinking that those are much more interesting. And he makes a mold and he puts his cell cultures into the mold, and he grows a steak and serves the results to a restaurant that are very appreciative and very rich clientele who are very happy to pay for a much more authentic form of food. So this is obviously not a real steak, this is made of plastic, but what's nice about making objects that bring a piece of the future to the present is that rather than writing a paper or a magazine article or a journal, the images transfer very, very quickly and very easily, and people can look at it and understand it as they would understand a regular product. So when we go to the supermarket, we look around and we look for things that we might want to eat, we might look at things we might want to buy and incorporate into our lives. And as a designer, you can create an imaginative object and present it exactly the same way a product today is presented. And people get to think about these new ideas and these new technologies in the way that they used to, in the way that they're used to making consumer choices or choices about what they buy and what they'd like to eat. And that means you get to ask much more interesting questions. So rather than people saying biotechnology is good or biotechnology is bad, you get to say things like, how much did it cost? Does it look appetizing? Would I want to eat it? And those are very much more interesting questions. because it's in the language of products as well, it sits next to the product. So here's an advert for Head and Shoulders shampoo, and here's a piece of meat from the future. And it's very nice that they exactly the same sort of image. The next project I'd like to talk about is one called Inchromite, and this is synthetic biology and the same style of as Alexander's been talking about. It's a collaboration between myself, another designer called Daisy Ginsberg, and University of Cambridge in Britain. And these are students age 18, 19, quite young. And this is the summer of 2009, and they're learning synthetic biology for the first time. So they're engineering, maybe trained to build bridges. Some of them are computer scientists. Some of them are chemical engineers. But they're all sitting in this workshop and they're being told, very much like you, what synthetic biology is. And by the end of the summer, they created this, which is a bacteria that creates a purple-colored pigment. That pigment didn't exist in that bacteria before. They engineered it to produce that pigment. So normally this bacteria is a sort of colorless or maybe slightly yellow color. But now you can see it producing a very strong kind of violet color. And that color has been taken from a gene from a different organism and implanted into this E. coli bacteria. Daisy and I sort of branded it for them. We called it Inchromite as a kind of pun, which is very popular in the field of synthetic biology. But they didn't only produce the purple color, but you can see that they produced different kind of reds, different yellows, greens, and this purple. Almost all of the colors of the rainbow. And these are students that learnt this for the first time and then spent 10 weeks doing this. And none of these organisms existed before they started. So it's quite interesting to see that while something conceptually simple like the hamburger made from meat cells costs $100,000 and has taken many, many years to produce, some students can visit a lab for the first time and produce organisms that never existed before and do things that they've never ever done before. And it really shows you that synthetic biology in this style, genetic engineering, is actually within the grasp of most people to be able to do. And you might not think that at first glance. You might think, oh, it's too complicated, it's not for me. But actually, a lot of this engineering can take place in the morning. It's the design that's the complex part. I'd like to show you a brief animation as to why the students wanted to produce these coloured bacteria, because you might be wondering, oh, why is coloured bacteria useful? So in their own voices, there's a short film lasting about 2 minutes that explains why.
And joining us now is one of the winners of the iGEM competition. Welcome to Science Friday, Ms. Bowen.
Hi, thank you so much. Well, I'm part of the Cambridge 2009 iGEM team, and our project was called Inchromite. And what we were trying to do is to improve bacterial biosensors. They're bacteria that can tell you the concentration of a pollutant in water. And they can do this because inside them they have a detector. So we developed 2 different parts, the sensitivity tuner, and this actually tells the detector when to turn on and when to turn off. So you have control over what level of the pollutant you're detecting.
And how does the bacteria show that it's on or off?
We use something called a color generator, which means that bacteria changed color when the detector got switched on.
Wow, so they light up in a different color.
They actually change color visible to the naked eye.
So let's say if you put a swab of the bacteria in a polluted river, bacteria would just change color.
Yeah, exactly. So you probably want to put a sample of your water on a bacterial site, maybe not the other way around.
Well, how would you envision something like this being used other ways in the future?
So what they wanted to design was a biosensor. So it's very simple. You have a test tube full of this bacteria, you put some water in, and the water changes color if it's got a toxin in it. And generally the toxin that they were aiming for is arsenic, which is very, very difficult to detect, and is a very widely, it contaminates a great area of the world, especially in the Asian subcontinent, India and Bangladesh, and places like that. And the idea would be that you produce very simple, very cheap, maybe credit card sized devices that you can distribute widely, and people can test their water to see if they're drinking too much arsenic, and maybe filter the water accordingly. But as well as that idea, Daisy and I were very interested because you have bacteria that produce colour, so what else can you do with them? And we decided to come up with a range of speculative products. We came up with about 7 or 8. And I'm just going to show one of them. And it's called Scatterlog. And it's a product that can be bought in the supermarkets in the year 2049 as a yogurt drink. So you buy it just like Yakult or Danone or something like that. And suspended in the drink are bacteria, because all yogurt has bacteria in it, but these are Inchromite bacteria. And when you ingest it, they're going to colonize your gut. So everyone has bacteria in their bodies already, and the bacteria in this drink here would just add to that. And these bacteria, they're going to sit in your gut, and they're going to stay there, and they're going to be monitoring for different diseases. So every disease has a chemical marker called a biomarker, and these bacteria are constantly looking for those biomarkers to see if you have something. So perhaps the biomarkers for litis or salmonella or a stomach ulcer or something much more serious, maybe like cancer, the bacteria will be constantly looking for them. And if they detect one of those biomarkers, then they're going to start generating a very, very strong colour. But of course this colour is going to be inside you until you go to the toilet, and then the colour will be outside you. And you'll get a very strong indication that something might be wrong, and you should go and see your doctor and maybe get yourself checked out. This is another project. It's also synthetic biology, sort of. What you're looking at here, I'd like to is an experiment of the team of scientists I was working with. Could I have a very, very quick show of hands as to who thinks they're looking at a living organism, so something that is alive? Maybe put your hands up. Living? No one? One? Okay. Who thinks this is a non-living organism? Who thinks this is just something that's not living? 2, 3, 4, 5. Okay, you're a scientist, that's okay. So what you're looking at here is a very, very simple chemical reaction. So this is not a living thing. It's not growing because it's alive. It's not a cell of any sort. It's just a simple chemical reaction between 2 chemicals that forms these tubes. But the scientists I'm working with are very interested in these because what they're interested in is building much more complex chemistries than are normal to create things which look live, feel alive, simulate some of the properties of living systems. And they feel that if they do this, they'll be able to for long enough, they'll be able to create a living organism from chemistries that we don't find in nature at all. So not DNA, not proteins, not fats, not amino acids, nothing like that, but completely different chemistries. And the reason why they want to do this is they want to understand what is life, what makes the difference between something living and something not living. And that's been a very, very difficult question to answer for many years. So if you look at the natural world, you have viruses which can last thousands of years, dormant, don't reproduce by themselves, and yet we kind of think they're alive, or at least alive at some point, because they do form part of kind of the ecosystem. So they have this question, and they're going to produce these chemical systems for many years until they reach something that everyone will look at and go, yeah, that's definitely right, it's reproducing, it's a little bit. But as a designer, since I think it's interesting, this collaboration between design and science, I looked at this from a slightly different angle, because I looked at their experiment, not as science, but as a design, where you may produce a prototype and see, oh, it's not very good, and so you improve a few things and you produce another prototype, and you think, well, it's slightly more alive than the last one, but not really. And you do that again and again and again until you reach something which you're happy with and you're comfortable with, and you think you've reached the end of your project. And I think anyone who's gone to design or art school will be used to that process of iterative design and improving on what you've made the day before. And if you look at their experiment in that way, and you imagine that they're successful in 10 or 25 years' time, then it's very interesting, because they will have proven that there is no clear distinction between non-living and living things. They will have proven that there's a continuum, a scale between non-living and living things, because the thing that they made at the beginning of their experiment will be maybe 5% alive. And the things that they're making towards the end of their experiment will be maybe 95% alive. And everything in between will prove that it's possible to make something that's half alive or 3/4 alive. And I think that's a very, very interesting philosophical point. And they haven't even had to do that experiment, but by thinking about their experiment from a design and iterative design perspective, almost come up with a new idea of what life might be and how you might define it. So what I wanted to do for my project was to define what the scale might be in a speculative way. So before the experiment is really taken off, just start at the beginning and imagine what the scale might look like in the future. So how in the future might we measure the difference between non-living and living things? And so what I did was I kind of like the workshops that we're probably going to do today, I just took a piece of paper and some pens, and I asked the scientists I was working with to mark on the scale between dead and alive the properties of the living or a non-living thing and put them in an order that they could use. And the reason why I show this is that it was very, very difficult for them to do. They couldn't do it. It was a philosophical mess in their heads. So up here they have written the word light bulb, solar cell. Down here we have stupid, passive. Up here we have intelligent and active. Okay, there's something there. There's something here about learning and adapting, but it's a mess. There's nothing very easy to understand from this drawing. Here's another one where this team of scientists felt that the most important thing about being alive, the first step on the scale was eating, and the second step on the scale is pooing. So again, not very interesting sort of ideas. And then I changed the question. So I went to talk to another scientist in the group, and I said, okay, I'm not going to ask you this question because no one's been able to answer it. But what I am going to ask you is just to explain to me what your research process over the next 10 years is going to be. What are you going to do first? What are you going to do second? What are you going to do third? So just tell me what you're going to build and make. And he said, oh, that's easy, okay. We'll start with creating the inorganic building blocks. So we'll work on creating not proteins, but the equivalent of proteins and DNA and things like that. And then we'll work on energy-releasing reactions. So we'll create a chemistry that releases energy. And then what we're going to do is cycle that energy. So we'll have a circular reaction where the products of one reaction feed into the next reaction. It goes in a cycle. And then what we're going to do is we're going to put these different chemical reactions inside membranes, so mimic the sort of cells and the way the cells divide and subdivides chemistry in that way. And then we're going to apply selective pressure. So we're going to make the cells do something, and the ones that do it successfully will survive, and the ones that don't will kill and evolve them. And that will create natural selection. And at that point, we think that what we'll have is a living system. I said, oh, that was much easier. Thank you very much. Now we have our scale between non-living and living things. So I tidied it up a bit and made it a little bit prettier. And then I made a film. And this film is, again, 2 minutes long. And it's going to tell you about the history of the emergence of life in the pharmaceutical industry. So imagine that we're in the year 2025 and you're watching a film made by Pfizer or Novartis or one of those big pharmaceutical companies about how life evolved in their industry.
For over 200 years, the compressed tablet has been the preferred way of introducing medicine into the body. But when the tablet was replaced by the chemical cell, medicine changed, and so did our understanding of living things. The first true chemical cell approved for use in the healthcare industry is a simple construction. It is manufactured by forming an inorganic membrane around particles of a given drug compound. Only under specific conditions does the membrane become porous, releasing small amounts of the drug where it is required. Advances in metabolic engineering led to the second generation of chemical cells. Lying dormant inside the patient for several weeks, they monitor the condition of their surroundings. When triggered, they are capable of manufacturing a specific drug, releasing it as and when it is required. The third generation of chemical cells are equipped with the autocatalytic machinery necessary to replicate themselves. Useful for treating chronic conditions, they maintain a constant population in a patient over extended periods of time. By reproducing in pairs, the fourth generation of chemical cells bear offspring that combine the metabolic processes of both their parents. Often used when a patient's condition does not respond to a known compound, these offspring will produce novel varieties of drugs, some of which might have a positive effect. The fifth and most recent generation of chemical cells are characterized by one important additional feature. If these cells determine that the drug they are producing is ineffective, then they make themselves the target of the patient's immune system, which will treat the cell like any other pathogen, killing it. This ability to die subjects the chemical cells to a form of natural selection, allowing their evolution towards a more effective treatment of the patient's condition. The medicines described here represent key milestones in the development of chemical cell therapeutics, but they are also the increments on a scale between non-living and living things, the cellularity scale. This is used in the pharmaceutical industry to classify chemical cells for regulatory purposes. But we prefer to see it as the route to a new tree of life, the branches of which would doubtless bear the fruits of our future health and well-being.
So I'd just like to finish with this quote by the scientist Richard Feynman, which I think sums up the spirit of sciences like synthetic biology, but also the spirit of design and art and making and learning by doing. And it's very simple. It's what I cannot create, I do not understand. And I think both designers and synthetic biologists would take this to be true. I think that's why there's a good, strong collaboration between both types of people. Thanks very much.